So we all know that microorganisms are very tiny and microscopic. They're not easy to see. So they are so tiny, but still they cause so much harm and damage. So how can something so small cause that much damage? That's because they are usually found in groups, in plenty, or in large numbers. Now, the way a tiny microorganism quickly becomes that much is because of its growth. How it grows. It doesn't grow in size to become very big, but it multiplies in number. So that's what we're talking about today, the growth of microorganisms. So as I mentioned, microorganisms show growth by increasing in size and in number of cells, majorly. They don't really show growth by increasing in mass or in width. They also grow that way, but the growth we are talking about majorly is increasing in number of cells, increasing in number of the microorganisms. Now, when the environmental conditions that are necessary for growth, such as food, adequate temperature, and humidity, when those conditions are right and are suitable for the growth, the growth will be hastened. It will be made faster. So, increase in the population size of microorganisms is due to reproduction. It's only by reproduction that we can get the number of microorganisms to multiply or to increase. So under favorable conditions, microorganisms can reproduce asexually by binary fission. Not all microorganisms reproduce asexually by binary fission. There are different other ways they reproduce, but under favorable conditions, the ones that reproduce asexually can reproduce that way. And a bacterium will grow to a maximum size, the size that it's supposed to grow to. Then it undergoes binary fission and divides into two daughter cells. So this process is repeated. The bacterium grows to a maximum size. Then when it reaches that size, it divides, it goes through binary fission, forms two new daughter cells, then rinse, repeats. The process is repeated all over again. Now, a species that is capable of rapid growth can divide every 20 minutes. So imagine if we are talking about if there is growth every 20 minutes. Imagine in an hour, how much multiplication would have happened. So that's how quickly microorganisms grow. So imagine in 5 days, in 10 days. So this 20 minutes that we gave as an example, we call it the time interval, or the time interval between. So this 20 minutes is the time interval between when the organism or the microorganism reaches maximum size, then doubles or divides or forms new offspring. So this time interval, we call it doubling time, or we call it generation time. So different microorganisms have different doubling times or different generation times. So generation times, as we've mentioned, or generation time or doubling time, varies from species to species, and it greatly depends on the environmental factors, such as the availability of food, how much food is available, then the availability of space, of temperature, and other environmental factors. So this doubling time or this generation time could be quicker depending on how favorable the environmental conditions are. So under ideal laboratory conditions, the generation time for Escherichia coli, that's a bacteria, is about 30 minutes. So when we place Escherichia coli under laboratory conditions, ideal laboratory conditions, the generation time is about 30 minutes. So theoretically, since the generation time is 30 minutes, one E. coli, when we say E, we mean Escherichia, so we're shortening it. One E. coli could divide into two after 30 minutes. So after every 30 minutes, one could divide into two. So, and so on, and so on. So the doubling is at intervals of 30 minutes. 30 minutes. So this type of increase is exponential, or we could say it is logarithmic. So it's an exponential increase. Every 30 minutes, it doubles. Every 30 minutes, it doubles. So in that, we are just saying theoretically, under ideal conditions. So this type of growth is the ideal growth, the growth that should be observed normally, under normal conditions, if everything is the way it should be. But most of the time, we don't really have an ideal condition. So we have the ideal growth, and we also have the real growth of microorganisms. So now, still talking about this ideal growth, the ideal growth, we can measure it, or we can view it by having a curve or a graph. So we could have an exponential curve. So what we do is we plot the number of bacteria against time. The number of bacteria against time. So how much bacteria is being formed each minute. So we can get a curve, we get an exponential curve where we plot the number of bacterial cells against the number of time, or the time in minutes or in seconds, the time. We plot that against each other and we get a curve. So we could see that on the screen now. There's a curve that is formed. So when we plot the number of bacterial cells against the time in minutes, we could put the number of bacterial cells on the horizontal axis and the time on the vertical axis. Also, if we plot the logarithm, if we take the logarithm of the number of bacteria, log to base 10, for example, if you take the logarithm of the number of bacteria formed, we could get we'll get a straight line graph. So if we have the logarithm of number of bacteria, then we plot it against time. So we'll have a straight line graph, So that is under ideal growth. So that's the way growth looks under ideal conditions, under ideal laboratory conditions. In actual growth or in real growth, the growth can be observed, growth of E. coli now that we're using as an example, by inoculating bacteria, the E. coli, into a liquid culture medium. And we measure the growth at regular intervals of time. So we can take the growth of E. coli, the way we measure it is we introduce it into a liquid culture medium. Then we measure this growth at regular intervals. So the growth is divided into different phases. We have an initial growth phase, which we also call the lag phase. Then we have an exponential phase. Then we'll have a stationary phase. Then we also have a phase of decline. So let's talk about these phases and how we can observe them. So when the bacteria, the E. coli, is introduced into the culture medium, it takes some time to adjust into its environment. So that's what we have, the initial slow growth phase. That's the initial growth phase that we also call the lag phase, that is slow. So the bacteria takes some time to adjust to its new environment. It doesn't just start multiplying at a quick rate. It takes some time. So that time, the growth is slow. So after that, the growth begins to proceed at an exponential phase. It begins to increase in speed. The speed increases very fast. So the bacteria reaches the maximum rate it can multiply. For example, when it was adjusting, every 1 hour, a new bacteria doubles. When it reaches the exponential phase, it could reach its maximum rate. For example, that I've mentioned E. coli is about 30 minutes. So it will reach that maximum rate in the exponential phase and start doubling every 30 minutes instead of 1 hour. So it becomes faster. So, however, when this size starts increasing at a very fast rate, then the population obviously starts to increase. The population would get so much that there will be competition. So obviously we now have a very large number of these bacteria. So there will be competition for food and there will be competition for space also. So this competition will lead to slow growth rate. So the rate of increase will reduce. It will reach a decline phase. Instead of increasing now, it will start declining. So after this, obviously the population starts to decrease. After it has started increasing at a very fast rate, it now starts to decrease because it's just like you place too many dogs in a particular place, then you put food for them, maybe you lock them in a room. Obviously, the stronger and faster dogs will quickly get the food, so they will keep increasing. So just the same way, the bacteria start to compete because they have multiplied up to a stage that they are so many. So there's a competition. So the competition for food and space now results in decline. So the growth rate stops until it reaches a state that it becomes stationary. Then the population starts to decrease. Some begin to die off. So when we reach that stationary phase, the waste, since at the stationary phase, the growth is no longer occurring at a fast rate. So we start having toxic wastes increasing instead of the food or space increasing. So this will cause the cells to stop dividing. Instead of dividing every 30 minutes and producing new ones, the cells have to stop dividing because there's now much toxic waste and there's a decrease in food that they can feed on. So these cells that are no longer dividing start to die off, and the growth rate reduces and the population reduces. So we can also represent this on a graph with it's a curve. So we start from the lag phase where there is no growth. Then we get to the exponential phase where there is maximum growth. Then the lag phase, there is a little growth, but the growth is very slow. So the lag phase, we have slow growth. Then we have the exponential phase where we have the maximum growth. Then we reach the stationary phase where there is zero growth before we get to the phase of decline where the population starts to reduce and the growth stops. So that's that for growth of microorganisms. So how can we measure the growth of microorganisms in the lab? Now, the way we can do this, we have multiple methods. So let's start with the first method. One method we can use, so these are the steps. We have a bacterial sample that is inoculated into a nutrient broth. So we inoculate, or we introduce this bacterial sample into a nutrient broth. So this nutrient broth is just a liquid culture medium, but we make sure it is clear, that we can see through it, that we can see through the containers, we can see what is going on inside the culture medium. So when we introduce this bacterial sample into the culture medium, obviously the next thing that starts to happen is that the bacterial population starts to increase. So when the population starts to increase, the clear medium, we introduce into a clear medium, visible, something that looks like water, a clear culture medium. So when this population of the bacteria starts to increase, the liquid becomes cloudy because of the increase in the bacteria. The bacteria starts to cloud the clear liquid. So it's just like you start pouring dirt into water, so it becomes cloudy, it becomes dirty. So it's just like you introduce the dirt into a clean sample of water. So the liquid medium, the culture liquid medium, starts to become cloudy, or we could say it starts to become turbid. Turbidity increases. Now, the progressive increase in turbidity indicates the increase in the number of bacterial cells. So we could measure the turbidity of the liquid medium, and that as the turbidity increases, that could show us the level at which the bacterial cells are increasing. So that way we can measure the growth of the bacterial cells. So the instrument that we can use to measure the growth of microorganisms is in the lab. We could use the spectrophotometer. So this instrument can measure the amount of light that passes through a liquid medium. So that's what the spectrophotometer can do. It can measure the amount of light that passes through a liquid medium. So as the turbidity increases, as the bacteria is growing and increasing in number and in size, the turbidity of the liquid medium also increases. So that means the amount of light that can pass through it is just like you have a clear bottle of water. The light can pass through that clear easily than a dirty, maybe brown liquid. So that was what we mean by turbidity. So the amount of light that can pass through the medium decreases. So that way we can measure the turbidity at regular intervals. So as the turbidity is decreasing, the way we can know the way turbidity is decreasing is that the light that is passing through will start decreasing. So the turbidity, we can know the level of turbidity, and we can use that to measure the growth of bacterial population. Now, one problem with this method is that both living and dead cells are included in this calculation. So the waste products that are not part of the actual living bacteria that we want to measure or count will increase the turbidity. So we may not really get an accurate number or an accurate calculation of the measurement of the growth of the bacteria. Now let's talk about another method of measuring the growth of microorganisms. So what happens in this method is that small samples of bacteria are taken from the nutrient broth. So after we've introduced a bacteria into a nutrient broth, we take small samples of it at regular intervals of time. Now, as we take the sample, we dilute it several times using a method called serial dilution. So we dilute it several times. So we dilute it up to a particular level. Then each sample is then inoculated onto a nutrient agar medium in a petri dish. Then we incubate it. This one will be a solid nutrient agar medium. Then we introduce that sample, we inoculate it into that nutrient agar, then we incubate it. So with that, we could look into the petri dish and count the number of colonies that are formed on each petri dish. Those colonies that just shows the number of bacteria that is formed on that nutrient agar medium. So we can measure the growth of the bacteria in that way. So as each colony is formed by the multiplication of a single bacteria, so as the bacteria is multiplying, a new colony is formed. So as these colonies are formed, we can use the number of colonies to know the number of living bacterial cells present in the diluted sample. So from that, we can get the actual number of bacterial cells in the original sample. We can calculate it after using the diluted sample. Because the reason why we are diluting it is just to kind of reduce the population so that we can easily count it. It will not just blow out on the nutrient agar. We could count it. These are the colonies formed and calculate that. Use that. We can add that and calculate the original or the actual number of bacteria in the original sample. Now, to count the bacterial colonies accurately, the colonies must be spaced out. So using serial dilution, as I mentioned, ensures that we get at least an accurate sample with countable colonies. So the serial dilution ensures that we make sure serial dilution, what I mean by serial, we dilute it several times. We dilute it about 5, 6, 7 times. So doing that, or maybe you could say 10, you could do 10, 20 times. So when you do that, that means we'll get at least one petri dish, one plate that has a number of countable colonies. So when we have up to 30 to 50 colonies in a particular plate or petri dish, that's an acceptable number. So we can be able to count 30 to 50. That means you've done a successful experiment. Now in this method, the difference between the first method and this method is that in this method, only living bacterial cells are counted. So we can only get the number of living bacterial cells from this. We don't count dead bacterial cells, so unlike the first method. So that's the end of today's lesson. See you next class.